Good afternoon, and welcome to the Sutro Biopharma Virtual Deep Dive Research Forum, Next Gen Innovations in ADCs. At this time, all participants are in a listen-only mode. Following the formal remarks, we will open the call up for your questions. Please be advised that the call is being recorded at the company's request and will be available on the company's website for at least 30 days. I would now like to turn the call over to Bill Newell, Chief Executive Officer at Sutro Biopharma. Sir, please go ahead.
Thank you, operator. Good afternoon, everyone, and thank you for joining us. With me on the call today are Dr. Trevor Hallam, our President of Research and Chief Scientific Officer, Dr. Kristen Bedard, our Vice President, Discovery, Dr. Shabir Anik, our Chief Technical Operations Officer, and Dr. Venkatesh Srinivasan, our Senior Vice President, Process and Analytical Development. Before we start, I would like to remind you that today's call will include forward-looking statements. These forward-looking statements are based on Sutro's expectations and assumptions as of the date of this call, and Sutro's clinical development programs, future results or performance could differ significantly from those expressed or implied. Please refer to Sutro's filings with the SEC, including our most recent 10-Q, for information concerning factors that could cause Sutro's actual results to differ from those expressed or implied on this call. As a company, we've been looking forward to this opportunity to share with you our most recent pipeline and platform advances, which include what we believe could be truly transformational next-generation therapies for patients with the most challenging to treat cancers. Our team has been hard at work developing additional novel therapeutic candidates and even new therapeutic categories, which Trevor will share with you in greater detail momentarily. On the agenda today, we will share an overview of STRO-003, our newest wholly owned product candidate, a novel antibody-drug conjugate designed to target ROR1, which is broadly expressed across a wide range of tumor types. A deep dive discussion of platform and our differentiated approach to generating impactful therapies, including immunostimulatory ADCs or iADCs. Our process development platform, which enables all of our innovation and for which we are constantly innovating. And our manufacturing strategy, which includes an in-house fast-to-clinic supply and a robust path to commercialization. Following the presentation, we will open the call to questions, at which time Trevor and I will be joined by our research and CMC leaders, Drs. Anik, Srinivasan, and Bedard, who have been instrumental in supporting the efforts that have led to today's forum. To kick off the discussion, I'd like to provide a brief overview of our pipeline. As this slide indicates, Sutro now is responsible for 6 product candidates in clinical development, and STRO-003 will be our 7th. We have assembled a diversified portfolio for product candidates for hematologic malignancies, including CC-99712, our BCMA targeting antibody-drug conjugate partnered with BMS, and STRO-001, which is targeting B-cell malignancies. We've also assembled product candidates for solid tumors, such as STRO-002, which is in dose expansion phase of phase 1 clinical development, and we're excited to be moving that molecule forward into a registration-directed study. More will be spoken about that later in the year. We also have M1231, partnered with EMD Serono, and most recently MK1484, partnered with Merck. In addition, our platform has enabled VAX-24, a 24-valent pneumococcal conjugate vaccine by our spin-out Vaxcyte, which is in phase 1/2 development as we speak. As a little bit of history, I want to remind you that beginning in 2018 through today, Sutro has averaged more than 1 IND filing per year from products emerging from our platform technologies. And we look forward to adding to that with a STRO-003 IND and eventually our first iADC IND which may well come from our collaboration with Astellas. From here, I would now like to hand the microphone over to Trevor for additional comments about our pipeline and then to begin to walk you through what we've done. Trevor, over to you.
Thank you, Bill, and good afternoon, everyone. It's a pleasure to talk to you on our latest innovations and where we believe our emerging portfolio is going to go. Just while we're on this slide, as Bill just mentioned, we have 6 different clinical studies going on so far, clinical candidates in development, 4 of which are ADCs, one a vaccine conjugate vaccine with vaccine, and the other a cytokine variant. The one thing they all have in common is that they're all designed and manufactured using this unique platform that Sutro has the advantage of. And this is a cell-free synthetic biology platform, effectively, where we can design proteins incredibly quickly, going from DNA to protein in 12 hours. That allows us to place very precisely and with varying number of precise conjugation sites in these molecules. So the common thing about all of these different biologics is the precision with which they're designed and the specificity and fidelity of their conjugation sites. This ability to design fit for purpose is really, I think, a unique differentiator for Sutro, allowing us to conjugate very precisely and to completion to result in homogeneous molecules in a drug, which is quite unusual in complex biologics to be able to get that precise. And also our ability to conjugate at sites which are not pre-designated by natural amino acids like cysteines or lysines, but where we have the flexibility to roam around the protein to find the best performing site. So for ADC, that's all about a really efficient site that's optimized to be able to deliver a cytotoxin to a tumor cell. For conjugate vaccines, it's all about the efficiency and precision of conjugation to allow as many as 24 different strains of pneumococcus to be represented in the tumor and retaining the balance between a full protein and those things that are conjugated to it to optimize immunogenicity. And for a cytokine variant like MK1484, there are a number of advances where we're able to directly conjugate to get best performance out of those cytokines. So without further ado, I'm going to talk you through our next up, which is STRO-3, this ROR1 ADC Bill just mentioned. And also then for the rest of the presentation, I'm going to talk about this inherent integrated combination of new processes and new products that come together in their design to give us, for increasingly complex biologics, actually an increasingly simple, straightforward, and efficient manufacturing solution. So I'll get going with STRO-3. If I can find the toggle for the next slide. So STRO-3 is a ROR1 targeting ADC. Why are we so excited about ROR1? Well, for one thing, the ROR1 biology makes it an attractive ADC target in that it has an active role in cancer progression and is expressed in tumor and tumor-initiating cells. Perhaps the more important thing, though, is that it's that specificity of expression on tumors and its restricted expression on normal tissue means that there is low potential for on-target toxicity. So the second thing that's really quite interesting of ROR1 is its broad expression on many different tumors. There is an expression of ROR1 in wide hematological malignancies, as well as a broad solid tumor expression as well, and that includes large indications such as non-small cell lung cancer and breast cancer. So the trick to getting this is to getting an ADC that's really performing as best as can be with the relative component parts, is to recognize that a lot of these tumors do express ROR1, but there is a combination of heterogeneous expression across the different indications and a fairly low copy number too. So that favors therapies that are very potent in their ability to cause tumor cell death with low antigen density or low copy number of the target antigen, in this case ROR1, you have to make sure that if for an antibody drug conjugate, it's going to deliver those cytotoxins in sufficient quantity on each individual tumor cell to tip that over into a cell death. And of course, that's more complex with low antigen density, needs greater efficiency in the design, and that's where our site positioning comes in. But it also potentially needs payloads, so mechanisms of cytotoxicity that are really very potent. So this favors the more potent DNA disruptors rather than the more common tubulin inhibitors as a potential mechanistic target. That doesn't mean to say that tubulin inhibitors can have an effect. In fact, there's some pretty reasonable clinical validation for both efficacy and safety in hematological tumors with an MMAE-derived ROR1 ADC, and which is also being expanded into solid tumor indications. But we feel certainly there's an opportunity here to engage with some of the newer mechanisms that have been targeted in ADCs and to be able to get to it. Now, potent DNA disruptors like cross-chain chelators like pyrrolobenzodiazepines can be a challenge from a tolerability perspective, although are very active even on cells that are not dividing rapidly. So we've gone with an exatecan class, which is Daiichi Sankyo has had great success with drugs like Enhertu. And we think this would provide that sort of ideal ability to tackle cells that are dividing slowly. The exatecan class has got good bystander effects, so potentially drug that's released in the antigen-expressing ROR1 expressing tumor cells, that exatecan can actually also kill surrounding cells. But it's not as potent as some of these things. So the tolerability, which Enhertu enjoys, is very attractive in being able to dose up. And what will finally be important, we believe, is that every internalization event has really got to count and deliver the optimal amount of payloads because of the low antigen density. And so we've now developed technology with our site-specific precise conjugations that can load DAR8 and above by positioning non-natural amino acids, and we'll talk about that technology in a moment. So this just is a prevalence chart across many different tumor indications, and the highest prevalence being at the top with mantle cell lymphoma. And you can see that the prevalence can be quite variable depending on clinical indication. And in large commercial opportunity like lung cancer, you'll see towards the bottom third of this table in lung cancer, that prevalence can vary quite considerably from somewhere north of 45% to around 90, 93, 94%. Now, in addition to prevalence, which is really the percentage of samples that prove to be positive for ROR1 in these indications, this chart also grades them in terms of percentage highs. If you were to look at prostate cancer, for example, that's a 90% and above prevalence. It's only 15% high by grade. That relates to the antigen density, and that's a critical number for ADCs in that you want a number of binding and internalization events to deliver optimal amounts of payload. So the lower that number is, the more potent the warheads, the more efficient the antibody drug conjugate has to be. So with the design, this is why we've designed a DAR8 site-specific exatecan class to go after a broad range of clinical indications. This slide shows the design of STRO-3. I think this is the first site-specific DAR8 ADC in that these sites are chosen for optimal performance, not just for stability and PK, but also the most efficient way in which we know that this exatecan payload is going to be released in the tumor or in the tumor microenvironment, in that those linkers are designed to work with these sites of conjugation to give really optimal release once bound to the antigen. So the targeted ROR1 epitope is clinically validated in that we know that epitope is expressed in a wide range of diverse hematological tumors, and we expect that to be the case also for solid tumor indications. The precisely positioned non-natural amino acid that we use as a proprietary parasitomethylphenylalanine molecule, which is tremendously efficient in conjugation. It goes to complete conjugation in just a few hours. So we can generate this DAR8 conjugation in several hours with very little molar excess, which of course is extremely important to cost of goods and manufacturing efficiency. We've chosen a different sort of linker from the fairly normal cathepsin B cleavable linker. We've chosen a beta-glucuronidase cleavable linker. These are several examples of these new linkers in clinical studies. They are thought to be more specific to lysosomal and endosomal space and can give a potentially more tumor-specific release, which has the promise of adding to tolerability. One particular improvement with beta-glucuronidase is that these linkers are designed and are not as liable to cleavage by neutrophil-derived proteinases like neutrophil elastase, for example, where some of the Cat-B linkers can be quite susceptible. And there are papers out there which link that susceptibility to neutrophil-derived proteinases to their own demise, so to potentially can cause neutropenia. That's not entirely proven, and of course, site of conjugation can make a big difference to that. In addition, the topoisomerase class have been known when paired with cathepsin B linkers, like for example with Enhertu and others, to have lung tolerability issues. And that is something, of course, we also want to avoid, the pneumonitis and interstitial lung disease seen in patients. So the exatecan walkout is a fantastic payload, we believe. It elicits that potent tumor cell killing and bystander activity, but it also sends up a flag from the tumor cell that's under stress and this so-called immunogenic cell death, with a lot is made of it these days, but it potentially is a very important aspect of this in that it sends up a flag to the innate immune system to go after the cell. So we believe these elements working together could potentially give us an ability to go after low-level expression of ROR1, deal with the heterogeneity through bystander effect of the exatecan, and also be really very tolerable with this new combination with beta-glucuronidase. So the first proof of the pudding, really, the first test was to put STRO-3 and challenge some patient-derived non-small cell lung cancer tumors by way of xenograft, and this is representative of 4 such PDX models which express low and heterogeneous amounts of ROR1 antigen level on that tumor cell surface. So if you look at this, what we're looking at, these are 4 separate PDX models, 4 separate tumors, which are representative of these lower level ROR1 antigen levels. And we're challenging them either with vehicle, with STRO-3, which is this beta-glu exatecan linker payload, or an alternative Sutro design, which is the same DAR8 capable antibody that against ROR1 that uses STRO-3, but in this case we've conjugated to a cathepsin B exatecan to see whether, as an alternative development candidate actually, but also to see which of these might develop best. And this is a great test of whether the beta-glucuronidase in the human tumor system is going to do the job. So as you can see in all of these, in dark green, you can see in black, you can see the vehicle, so that's the normal tumor growth in each one of these tumors. And in dark green, you see that 4 doses at 10 mg/kg a week apart are able to completely cause tumor regression, at least in 3 out of the 4 of these, but pretty significant responses in all 4. By contrast, the alternative design, the Cat-B exatecan in light green, does really well in 3 out of the 4. And the other one, you can see a little bit of growth on the second from left. So not quite as efficacious. And actually, that was not too much of a surprise in that we know that the active catabolite on the cathepsin B linker is slightly less potent than the active catabolite that is derived from the beta-glucuronidase sensitive linker. So all in all, the efficacy falls out the way we wanted it to, or we're hoping to, with the beta-glucuronidase seems to do a really fantastic job in these low-level expression systems. So And the next question, this is actually looking at a human breast cancer model with moderate ROR1 expression, and looking to see the effects of the DAR8 beta-glucuronidase exatecan system here. So what we're looking at here in dark green is the DAR8 beta-glucuronidase exatecan targeting ROR1. So that does very well. Now, in this particular instance, this is 4 doses of 5 mg per kg, so slightly lower dose a week apart. And you can see a very good response out to day 50 or so with those doses. In light green, you can see the DAR8 CAP-B exatecan for comparison, which is, again, less efficacious. And for those of you that are interested in what a DAR4 CAP-B MMAE linker payload would look like on a similar affinity antibody, that's in blue. So you can see, you do see some tumor growth inhibition, not quite as significant as the beta-glu DAR8 exatecan. Now, this slide demonstrates that actually the killing by STRO3 is ROR1 dependent. An important point, we believe, in terms of tolerability. On the left side, you can see a comparison between 2 cell lines, one ROR1 3+, so high level of expression of ROR1, and then the other one, a ROR1 negative, the MCF7 cell line. And the reason for doing this, in red, you can see the effect of STRO3 on the relative cell viability of those cells. So we incubate these cells for 4 days. We're looking at their viability in the presence of either STRO3 or an isotype control ADC. So an ADC that doesn't bind to anything human, but is loaded with the same DAR8 exatecans, and that does not kill the ROR1 positive cells. STRO3 does, and you can see the dose response curve there. The incomplete killing is because this cell line is heterogeneous, although those cells that do express ROR1 on average express quite a high level. There is a certain population that do not express. And then the last one in blue, we've actually shown you what STRO3 will do, but now in the presence of micromolar of an anti-ROR1 antibody. And that is there to completely block the ROR1 site to prevent the ADC doing its thing. Now, if you have a linker that's not very stable and that comes apart, you will still see killing in this sort of experiment. And on the right-hand panel of the left side is just to show that when we incubate the STRO3 for 4 days in a ROR1 negative cell line, we see no decrease in cell viability. So we conclude that this is indeed antigen dependent and a stable linker. On the right-hand side, we just show that, again, a 2 mg per kg, 3 doses a week apart, we see some good tumor growth inhibition in the ROR1 positive xenograft model, which we just showed you before. So it seems to be quite well behaved. It is antigen dependent, and the linker is stable in these models. So tolerability and safety, this is key. So STRO3 is comprised of a monoclonal antibody that targets ROR1, and we chose this particular antibody not least because it was stable, had good PK and all the rest, but also because it had cross-reactivity not just to non-human primates, but also to rodents. So this allowed us to do a full analysis with the different linkers, not only the linker payloads, rather, not only to check to see how tolerable they were in an off-target manner, but also whether there were any antigen-dependent target tolerability issues we needed to consider. And we were thrilled to see that STRO3 in rat at high doses, up to 60 mg per kg, we observed no neutropenia at all and no elevation of liver enzymes. So that was really extremely good. For non-human primate studies, we did a multi-dose non-GLP non-human primate study, and we observed no neutropenia at all or thrombocytopenia up to doses of 45 mg per kg top dose. No changes observed in white blood cells generally. So that was phenomenally good news too. Now, one of the things we did do, because of the experience with Enhertu and other ADCs using exatecan, exatecan payloads, it was to have a look to see what the liabilities might be for lung toxicities. And again, STRO-3 at 45 mg per kg showed no changes, no pneumocytes, no infiltrates, no damage at all in the lung, which would be indicative of a developing pneumonitis or interstitial lung disease with this beta-glucuronidase linker payload. In contrast, the cathepsin B linker exatecan, and you'll remember that the active payload with the cathepsin B, although it's the same exatecan class, the active catabolite is slightly less potent than our STRO-3 active catabolite. We expected that this construct would actually be slightly more tolerable because of that less potent active catabolite. In fact, that's not the case. With the cathepsin B linker, we also saw some lung findings which were consistent with developing pneumonitis and potentially ALD at that 45 mg per kg dose. So going from cathepsin B to a glucuronidase linker, we both increased the potency because we had a more active catabolite in there, but at the same time, we made it more tolerable than a conventional cathepsin B linker. So beta-glucuronidase was clear that was our most efficacious, our most tolerated, and therefore the widest therapeutic window with this particular construct. It should be noted, and I said before, that other Cat B linker exatecan ADCs, including those, are associated with significant rates of clinical pneumonitis and ILD, although drugs like Enhertu are remarkable, fantastic overall response rates in solid tumors. But this is a serious concern. So improvements on that could really send us to be considering best in class. So we believe STRO-3 enables a broad clinical development strategy because we've got efficient killing of tumors with low-level ROR1 expression and a very favorable safety profile. It's an expansive indication space, and so we'll be looking certainly to explore the fullest number of indications that we think makes sense. Of course, ROR1 is validated in both hematological and broad solid tumors. The opportunity is there. Topo-1s, however, have only really been showing compelling clinical validation in solid tumors at the moment, although there's no reason, I don't believe, that they shouldn't also do well in hematological tumors too. It's just that is not properly validated as yet. There certainly are DNA payloads of the pyrrolobenzodiazepine class, for example, that are finding great efficacy in liquid tumors. So we believe that the design appears to demonstrate impressive efficacy with potentially reducing lung and neutropenia tolerability. Haven't spoken much about that, but that is completely clear. So, and those things are associated with topo-1 class payload ADCs. So we look forward to moving that forward. Our clinical programs, our strategy is going to be exploring when we get into the clinic, both the prime indication space, but certainly for solid tumors, this looks like a really excellent candidate. Now, I wanted to move from there and talk a little bit more about these advancements and the impacts on our emerging research portfolio, and also talk about some recent news some of you may have seen with a collaboration with Astellas. So let's just talk through our platform as it is at the moment. So we've been developing the platform, the component parts of this platform and our manufacturing strategy to give us a number of different tactical component parts that we can bring to bear. We already know that Sutro's precision design, its ability to flexibly and iteratively choose best possible sites for conjugation outside of the normal constrained natural amino acid targeting sites, can improve efficiency of killing. We also know, so the conjugation sites do matter. We've also used our system to be able to balance the relative binding affinities within a bispecific antibody to actually improve specificity of the agent to go after carcinomas. And we've used the fact that epithelial cells are normally polarized, healthy epithelial cells are polarized. So they have membranes where proteins are expressed specifically on one side of the cell or the other. And of course, as the epithelial cell transforms into a carcinoma, that polarization is lost. So we can use that by designing a bispecific antibody that has to bind to one of each antigen to give it the sufficient avidity to internalize and kill as an ADC. You can use that specificity to sidestep some of the common tolerabilities that you would get, for example, with EGF receptor binding. And that's indeed something we've done with EMD Serona, and that's a clinical development candidate on EGF receptor ADC. We've now enabled the topo 1 inhibitor payload with new very promising cleavable linker chemistries, which promise, or we aspire, we hope, that when we get into patients, they're going to have better tolerability and retain such great efficacy. That DAR8 has been a critical element of getting there. That's shown well on other molecules. And we've managed to get to DAR8 and above in a homogeneous way and a site-specific, not compromised by having to go to existing natural positioning on the antibody, but finding new space. And then lastly, we've been evaluating how to use these sorts of technologies and our ability to make dual conjugates on the same antibody, which opens up the opportunity to conjugate two different mechanisms of payload. We need to be smart about this, choosing payloads that are mechanistically synergistic and it makes sense to deliver in the same temporal and geographical location is obviously the way to go. And we feel this technology is going to open the lid on some new therapies. These are going to be new modalities that we've not really seen before, which move us beyond best-in-class to first. So I want to talk a little bit about that some more now. So our platform then, this is a good summary, I guess, on the left-hand side where we're looking at different modalities, the ADC on the left, the bispecific ADC on the right, which I just talked about with the example being M1231, the MUC1 EGF. But in the middle, we have this dual conjugation opportunity. And we've got two little subscripts here. IADC stands for immunostimulatory ADC, and ADC squared, which is we're not going to reveal at the moment, but this is really targeting a targeting modality using dual conjugations, which is really focused on getting optimal ADC activity, but also doing something about resistance pathways at the same time to really potentially enhance durability of response and depth of response. So we have options at each time because we manufacture and design these things in such a fast and iterative way. We can put these things together and make some fairly late discovery choices on exactly what modality we want to go with. So let me walk you through. So on the right-hand side, these are actually a bit of a reveal on the antigens that are on slow simmer at the moment, or not so slow, but the ones that we're very interested in at the moment, tissue factor, PTK7, integrin beta-6, LIV1 and CEACAM5 are all active programs within Sutro. We're looking at these both as monospecific, so a single antigen targeting potential, but also as potential bispecifics in combining one with another. And that's our first choice. Which ones do we go? How do we pair that with the indication? Do we need that improved specificity by combining into a bispecific targeting agent, or can we go with a monospecific? So once we have that, then we have choices about how we weaponize these things. We now have a toolbox of linker payloads, non-cleavable tubulins, cleavable tubulins, and cleavable DNA targeting classes, including the exatecans, but also including PNUs. So in addition to that, we've also been developing a number of different immunomodulators, including STING agonists, TLR7, TLR7 and 8 joint agonists, and other mechanistically synergistic payloads. That we've already talked about the proprietary cleavable, non-cleavable linkers that we combine with all of these. So the question is, what do we want? We can actually go to select a clinical indication or an antigen of choice. We can decide whether to tighten that up in terms of specificity. We can then decide at a late stage, once the antibody is designed, what do we actually want to do in terms of conjugation sites and how do we want to put something together? What payloads do we want to combine? So I'm going to give you an example, which is the basis of the Astellas collaboration we revealed publicly late last month. an example of combining a cytotoxin with an immune stimulus to give us a longer lasting response. So we've termed this an immunostimulatory ADC, an IADC, and it is a dual conjugation of cytotoxin and immune modulator. So this is a great opportunity, a completely new modality, and I want us to think about this rather differently from an ADC. For an ADC, you're really interested in a precise targeted antigen and how can you get the optimal amount of killing of the tumor by targeting that single antigen. In this case, it might seem a minor adjustment to just add in an immune stimulus as well as the cytotoxic payload, but actually the concept is very different. What we're looking to do here is mimic some of the very old knowledge that bacterial infections can affect tumor growth. It goes back, I think, several hundred years, or at least as far as William Coley, who did some of the first bacterial extracts and put those into cancer patients and got some reduction in tumor size. And that was the whole basis, really, of immunotherapy. Of course, these days with the FDA approval of things like BCG for non-muscle invasive bladder cancer, this whole area of toll-like receptor agonists and pattern recognition responses has come to the fore. So we're very much looking at this from a conceptual basis of, wouldn't it be good if we could try and stimulate an anti-immune response from the tumor itself? So if one could disrupt the tumor, release tumor neoantigens, and equip that with also agonists that would mimic those innate, and stimulate those innate immune systems, well, could you set up an adaptive response that would give you a far long-lasting response? So we set about trying to do this with a cytotoxin, which we knew we could disrupt and release tumor antigens, but also the hemiasterlin that we did a concept molecule with, of course, is on a couple of our clinical development candidates, including STRO-2. And what we did here was we'd used that particular warhead because we knew it caused immunogenic cell death, which is a bit of a popular phrase these days, but there's an interesting phenotype. As the cytotoxin puts the tumor cell under stress, this tumor cell starts to engage as a very natural physiological mechanism of signaling to the innate immune cells it's under duress. And this is by calreticulin expression on the surface, release of HMGB1 and other proteins which are able to stimulate pattern recognition receptors. And then in addition to this, we wanted to put an immune stimulus, and we used a TLR7 agonist as a prototype and plant that in the tumor cell. So what we envisage happens in these circumstances is that you are injecting or effectively systemically administered an antibody which delivers both the cytotoxic payload as well as the innate stimulus, and that will release the antigens. It will cause an immunogenic cell death response, which preps it up. Innate immune cells will come in after those released tumor antigens and the debris from those disrupted tumor cells. When those macrophages and other cells chomp down on that to clear that debris, they will find an activator, and that activator will then stimulate those cells, send up the flare, the beacon, and that's what sends in the T-cell responsiveness and sets up the adaptive immune response. So what we're aiming to do here is set up a longer-lasting memory response. So does it work? So this is the construct of a systemic administered monotherapy that drives anti-tumor immunity. This is a prototype molecule. Let me stress that this isn't a development candidate, but it shows you what we can do and how we can do it. So the purple stars on this graphic are really the hemiasterlin cytotoxin, conjugated to the antibody in a very precise way. So we know something about this. This is a hemiasterlin tubulin inhibitor on a cleavable Cat-B linker. And in red, we've put in a TLR7 agonist, which is actually conjugated to the light chain using another Cat-B linker. The way in which we construct these is that we are able to make a prefabricated light chain with a non-natural amino acid, and then around that run a cell-free synthetic construct of the heavy chain, which contains a different non-natural amino acid. So we can get very precise conjugation in any stoichiometry we want on the same antibody. So a one-pot conjugation at the end of all this in manufacturing delivers the precise ratio of payloads that we've chosen to these specifically chosen sites, and is very high fidelity for those different sites for each payload. I'll come back to this a little later because this is the basis of a sort of modular manufacturing base. So when we look at molecules such as this, we can see here we have these are MC38. So this is a tumor, a mouse tumor. We're looking at mouse tumors in an immune competent mouse mice system. And then top left, you can see the vehicle. So that's the seed rate and how well the tumor grows very, very quickly and aggressively. And it's this MC38 mouse tumor is transfected with the human tumor-associated antigen that our iADC is targeted to. If you were to look at the ability of an ADC that targets that antigen on the mouse tumor loaded with just a hemiasterlin DAR4, so this is in blue, top right, you can see these are individual mice. You can see most do quite well after a single injection of the ADC. The ADC does reasonably well, but and some of them do very well. There are 3 complete responses in this group of 8. The bottom left, you can see if you do the same thing with the same antibody, now conjugated with 2 TLR7 agonists on the light chain, you can see there is a little bit of a delay in tumor growth, but there's nothing like the response you get with the ADC. When you combine both of these things in red on the same antibody now, so 2 TLR7 agonists on the light chain, 4 hemiasterlin cytotoxics on the heavy chains to give you a 4+2 iADC, now you can see that directionally it's going in the right way. We're getting 5 out of 7 complete responses in just after a single dose. Now, if you take those surviving animals and now you re-challenge with the same tumor that's on the right-hand side, what you see is that you cannot regrow that tumor. Now, is this because the iADC hangs around for a long while? That's 60 days out when these are re-challenged, these mice, and the naive animals are age-matched mice, showing that that tumor will grow. We know that that's not due to residual amount of the initial therapeutic there, because we know that if we try and re-challenge with the MC38 cells, which don't have the initial tumor antigen, the human tumor antigen transfected into them, we still get complete inability to grow those tumors. So effectively what we're saying is that there's likely a cross-epitope learning, a memory response now, an adaptive immune response, which gives these mice protection from further regrowth or re-challenge. Further experiments we've actually done show that this is dependent on CD8 cells, and if we deplete CD8 cells, these tumors regrow. So that's pretty exciting to understand, and directionally points to the fact that these combinations may work well for us if we can optimize them for efficacy and tolerability. Now, if we look at those tumors and we ask the question of what does compared with vehicle, what's happening to dendritic cell activation in the tumor, and you can see on day 1 on the left panel that the dendritic cells are activated by an ISAC, that's just the targeted TLR7 alone, but not by the ADC, and they are targeted, of course, by the iADC, which also has the 2 TLR7s. And if we switch across to the right-hand side, you can see that although that innate response, as you'd expect, is a day 1 event, it's an early event, we're not seeing any change on day 1 in the adaptive immune, so the CD8 component part. But you can see at day 5, there's a developing response in the CD8 T cells in the iADC population, not so much in the ISAC population of TLR7 alone. And indeed, if you look at the CD8 Treg ratio, again, that seems to be enhanced, which is exciting. Now, if you I like visuals. So if you look at the staining of the CD8 cells in these different tumors treated with these different treatments, you can see that the iADC population of CD8s has increased. So that's very promising. Just quickly trying to make sense of an iADC versus these other sort of modalities, you can see that we think it's pretty important to have both the antigen released to direct tumor cell killing, as well as the ability to prime and stimulate the innate cells in order to drive T-cell recruitment and this adaptive response. So from that perspective, I think this new modality covers the gambit, really, in the ability to be able to drive an in situ immunization from a systemic administration. The thing that's really different, I think, though, is that what we're now seeing is potential for bystander effect, not driven by a cytotoxin, which bleeds out into the surrounding tumor microenvironment, kills surrounding cells. This bystander effect is caused by the immune system. So you're giving it the 1-2 punch of both antigen as well as an innate activator, which potentially sets you up to get almost like what dare I say, vaccination type response in response to a systemic administration. And it's obviously personalized to that individual patient's tumor. So this is quite complicated, and clearly the reason why I'm talking to you about a concept molecule rather than a development candidate is that there are a lot of moving parts and a lot of things we've had to cover to get that combination precisely of the right levels of each component payload placed in the right way. So, but there's great promise in here, and this is the basis of a very exciting new collaboration with Astellas, who signed up with us to push these some candidates through to the clinic as quickly as we can. Now, I want to go from there and just talk to you a little bit in the final few minutes and talk about our product and process design and how the integration and relationship between the product design is integrated entirely with the process design, and how that then converts for what are seemingly really complicated molecules into a very measurable component part modular manufacturing base where things become quite straightforward as we go forward, where there's predictable manufacturability. So I'll cover the key aspects. So we effectively have an open system. If you think of a large organization with maybe 100 different products, each of those biologic products is manufactured in a separate cell line. It's like having 100 manufacturing solutions. What we can do is generate 100 different things with a much smaller number of component parts. The reason for that is we don't need a separate cell strain for every product because we make everything in our portfolio today with one cell strain, from which we derive an extract. And that extract, of course, is clean. There are no living cells there. It's just the raw transcriptional and translational apparatus which we're harvesting. That extract can be used for all of the products in our portfolio today and everything that you've seen in the future from what I've just described. So that ability to break open the cell and use that as an extract gives us the ability to stockpile the extract and use it at any time. It's 12 hours from a protein when you add DNA to this extract and support it with the reagents, energy, and amino acids, of course. And you can do that at any scale. So the link between design and what you will do from a manufacturing base is incredibly close. You're choosing from many different protein variants which are going to be a lead. It could be an antibody discovery, trying your new CDRs in a fully folded IgG for the first time as first synthesis. And you can pick already which ones are going to fold up well, behave well, very quickly. And so your choice of starting points, your choice of antibody leads is biased by how manufacturable it is, because it's the same exact reaction which we foresee even at 20,000 liters. So we've already demonstrated this transferability from microliter, 5 mil, 10 mil, 8 liter level expression in our research labs through to larger scale in our process development labs and up to a 1,000-liter single-use bioreactor in Sutro's own GMP facility in St. Carlos. And from that sort of scale, it's de-risking the further scale to 20,000 liters and beyond if we so wish. So this idea of an integrated synthetic biology enabled by the fact there are no live cells or membranes in there gives you an open platform for different modular or component parts to play in your design. So we've already talked about one example, which is the prefabricated light chain. which we can actually put in and then build the rest of the antibody around it, and that can change how you conjugate to it. But the way in which we build this, of course, we're designing aspects of this which carry through and are common in process development. So the fact that we always use the same strain means we always have the same host cell protein background. And so many of these component unit operations become very familiar and very straightforward, and same again every time we change the product. So I'm going to try and get us some through how we do this. Now, on the left-hand side of this diagram, we talk about how we design. So we're looking at patient needs, where the limitations are really, where the treatments are really limited. Where are there precedent mechanistic things we could try out really quickly? And of course, we're always interested in commercial opportunity as well as the medical need. But the speed with which we can put something together is really key, because that means we can make many versions of something to optimize. We can look at different things quite quickly and engineer them very fast as proof of concept things where we can scale quickly as well to be able to do in vivo pharmacology and efficacy determinations, as well as go fast into non-human primates. And if we don't like the look of something, we can redesign and go back again within 2 to 3 months. And that's really novel in terms of biologics and the year-long process it normally takes to get a stable cell line to equip those sorts of studies with decent quality and quantity of product. The continuity is key. So I mentioned that the whole cell-free reaction that we do at very small scale is exactly the same as we do at large scale. So because ADCs are complex molecules, ultimately the clinical profile depends on all of these features coming together, their attributes in a well-behaved antibody. Those attributes can change. If you have to design an antibody and you express it in a stable transfection, and then you migrate to a stable cell line in time, and then commit to all of the difficulties in conjugation, the quality control, and so on, that the spec of that molecule can change in time, and sometimes that can compromise what was originally started off as quite a nice research tool. For our system, we don't change it. It's the same system. So not only are we able to preserve the integrity of the product profile, but we're also able to see how we will get cost-efficient gains. We're able to see that what we choose to go forward with is actually biased for the ability to be able to manufacture, because it's the same system. So that seamless scalability from milliliter scale to thousands of liters, I think, is really key with no formatting changes. The second part I wanted just to call out was the rapid process development. So we know we can get a cell-free reaction which can be quickly deployed to produce new molecule within days. It's almost an on-demand system because we can stockpile that extract. And beyond that, we have a number of components we can bring to bear. So we have the core cell-free product agnostic manufacturing process, the extract and the cell-free reaction. We also have off-the-shelf reagents which are specific to this cell-free reaction to support it, as well as, and I've mentioned before, stockpiled GMP-ready prefabricated light chain. So where common light chains are used across different antibodies, we already have that component part. We can bring them in. Linker payloads, we're all used to thinking of linker payloads as components that we can switch and change across different products. But the modularized process options by selecting from these now is really interesting. And we can take pre-tested, pre-optimized versions of these component parts and combine them in new products without having to reproduce everything from scratch as you would in conventional manufacturing. Lastly, the standardized control strategy, because it's the same strain that's producing the same extract, we're able to actually produce packages to refer to by health authority for health authority review, which are familiar. I mean, they're predictable CMC packages using the same component parts. I'm having problems going forward here. Yeah. So I just want to give you one example, and this is very relevant to conjugation. Prefabricated light chain. Now, for 2 of our molecules in clinical development, we've shown that pre-manufacturing a prefabricated light chain that's common to both antibodies is actually quite useful because we can add it into the cell-free reaction. So the cell-free reaction itself is only making a heavy chain and placing non-natural amino acids for an ADC if they're positioned in the heavy chain. So just the wild type sequence, native amino acid only, prefabricated light chain. We're very familiar with having that as a component part that's prefabricated. We actually make it in an intact coli on the same background strain as from the extract, so they're not confusing or contributing to the complexity of the GMP process. On the middle one is another variation. So think common light chain, same prefabricated light chain, but now with a non-natural amino acid in it. So this is exactly how we're using ROR1. Again, it's on a common light chain, so we could take a number of different antibodies that use a common light chain and substitute this component part in with a non-natural amino acid, late-stage discovery, just to ask the simple question. So in this case, do we want a DAR4 or do we want a DAR6? So you could do that on the left-hand side, or do you want a DAR8? You do that by this middle process. And then lastly on the right, well, if we can do that, we can also use the common light chain, but now have already equipped it with a different non-natural amino acid for an orthogonal conjugation chemistry, which allows you to do, in this case, a 6 on the heavy chain plus 2 on the light chain, 2 different payloads. So that's how we think of putting things together using this modular production process. And to actually just add a little bit more complexity to it, you have to balance the 2 payloads. There's no point in having one very potent, the other not so. It's not going to work. It will, that molecule obviously will, one payload or the other will dominate. So the ability to do what we do, and part of our optimization process is exactly this. We can switch the ratio, in this case on the left-hand side, of red payloads to yellow payloads. There's a 3-to-1 ratio on the left-hand side and a 1-to-3 ratio on the right-hand side. So this can be done by modular components and done very quickly, also looking at different sites to actually end up with the right balance together with the chemistry inputs. And we've got a strong medicinal chemistry outfit that's absolutely critical to everything we do. Although we're known for our engineering, we're also a medicinal chemistry company that basically ties in synthetic biology and medicinal chemistry to make complex molecules like this. So this is another key aspect of the flexibility of our system. Now, lastly, I just want to touch on very quickly our CMC and supply chain strategies for both 2 important aspects of what we do, faster clinic and commercialization. So the first thing, we have experience in the clinic. We have our own GMP facility where 6 product candidates have gone through, 3 monospecific ADCs, a bispecific ADC, a cytokine bioconjugate, and a conjugate vaccine. So what we've been doing is establishing and doing a robust external supply chain for clinical supplies of these molecules. So, we have linker warhead process chemistry and GMP manufacture. We have conjugation established with external supplies that can do that and form drug product from drug substance for both ADCs and bioconjugates. We obviously are doing fill and finish, so drug product and vials, clinical packaging. And so our extract and custom reagents, which are the rapid, common agnostic platform, can be applied very quickly to antibody-based and cytokine product development and manufacturing. So in the future, now we've equipped ourselves with these processes and support, we can move quickly to the clinic. And lastly, a strategy to CMC commercialization. So Shabir Anik, who's on the Q&A panel, will, I'm sure, be happy to answer your questions if I don't do this justice. But what Shabir and colleagues have been doing is establishing a CMO network for all of the components that we need to deliver scalability, notably extracts and then the cell-free reaction itself, but also including things like the prefabricated light chains and custom reagents to support this whole thing. And really that scalability that comes with this CMO network is going to allow us to build an inventory, minimize risk, and to support clinical studies and commercial launch in the future. So we talked about the extract. We can try it down now. It's quite phenomenal that you can take a biomass extract and dry it down, add water, and get all the attributes back, all the transcription, translation, and the capability to generate energy in situ by feeding it, low-cost energy. The scalability that's now being put in place externally is 10 times the capability or capacity that Sutro's San Carlos facility has, which is great for us to enter phase 1 clinical trial material. But this scale will now allow us to do registration studies and commercial. And then GMP batches for the extract are in process and will be coming through in the last half of this year. For custom reagents, we already have large-scale GMP batches manufactured, and prefabricated light chain have already exemplified with large-scale GMP batches manufactured for at least 2 clinical trial candidates right now. And then the cell-free reaction itself, the production of the protein, we've selected a CMO with GMP batches on track for the end of next year. So with all of this in place, the supply chain should produce over 250 kilograms of antibody per year. So lastly, just to conclude, we have a number of unit parts of our design and process and manufacturing which are impressive. We know the rules. In other words, we don't have to do an iterative design of every component. We know where we're going now. We have rules set. We have go-to sites for conjugations. And that convergent optimization of product design makes everything more efficient going forward. The modular process development, I hope I've done some justice to, can accelerate our process development and the way forward, and to have a predictable manufacturing platform. So the other aspect of our manufacturing platform is that it's very simple to outsource and produce on demand. It's flexible, so it allows us to move our technology around the world, or to obviously to our partners, allowing them to control their own CMC schedule. But lastly, just to conclude, I'm really excited that we've been able to turn this platform now into something that can come up with new modalities, targeting combinations now, but targeting to tumors to reduce their combined tolerability issues, to really play into getting the immune system to go after tumors that are cold. In other words, introducing cytotoxic T cells into those there to equip that, and also to address other aspects that are presently being done in combination, but where targeted therapies could do so much better, and to deepen responses by targeting resistant pathways at the same time. So with that, I'll turn it over back to the operator for our Q&A session. Thank you for listening.
Thank you. And at this time, we'll be conducting our question and answer session. If you would like to ask a question, please press star 1 on your telephone keypad. A confirmation tone will indicate that your line is in the question queue. You may press the star key followed by the number 2 if you would like to remove your question from the queue. For participants using speaker equipment, it may be necessary to pick up your handset before pressing the star keys. Once again, to queue up for a question on the phone, press star followed by 1 on your telephone keypad. We'll pause for a moment to poll for questions. Thank you. Our first question comes from Roger Song with Jefferies. Please state your question.
Great. Thank you for taking the question, and appreciate the detailed presentation for all 3 and IADC and CMC. So I have a couple questions related to all 3 raw one. So the first one is the magnitude of the cancer killing also the raw one expression level dependence? Because I see the non-small cell PDX model seems not clear, works pretty well even in the low expression level. But have you done any other model, as a cancer model, and see consistent results?
If you could handle that, please.
Hi, Roger. Yeah, I mean, what we were showing there is relatively high dosing. In fact, when we ran these studies, we were having a real good look. This is at 10 mg per kg, 4 doses. So if you give lesser doses or lower doses, you do see less efficient killing, but still remarkably good. I think in terms of I tend to think of antigen dependency and antigen density as, as long as there's antigen dependency, then the antigen density is more predictable in terms of dose. So you either get there with dose or you get with the antigen density for a given dose. So I think there's an intricate relationship between the numbers. What we're really trying to look at, though, is whether clinically this is in the relevant place for efficiency of killing. So if you combine this slide looking at the antigen density itself being low but giving good killing, and you remind yourself that actually the killing is absolutely antigen dependent, then I think the rationale follows that, that the antigen density will have a relationship with what you're dosing. It just depends on whether you're going low enough in dose to demonstrate that. And from what I've shown you, I've not shown you that. But that's our expectation. So I'm not sure I answered your question.
No, I think that answered. If you go dose lower enough, you will see the difference. Obviously, if you go higher dose, basically everyone kills those cancer cells, regardless of the antigen expression level. Okay, I got it.
The things that confuse it, Roger, are if the linker is unstable and you've got a systemic play of active catabolite or payload that's acting systemically in at the same time as you're trying to get an antigen-dependent response. So with ADCs which are poorly behaved and are shedding active payload as soon as you dose, you're going to lose that because it's almost like a slow-release chemotherapy. This isn't one of those. This is a stable ADC. And so the rules are a little bit more straightforward.
Yeah, got it. I see that in the second slide, you see the no killing in the raw one negative cancer model. So that speaks to the stable linker. Got it. All right, so that's good. And then, so I know you are just announced the candidates, maybe a little bit too early to talk about a clinical plan, but just broad stroke, what will be your timeline or the plan for the R&D filing and the potential clinical like indication in the study?
Roger, this is Bill. I'll let you know that this is a target that we're excited about because we believe there's tremendous potential not only in hematologic malignancies but also in solid tumors. As we're moving through the IND-enabling work and the CMC processes, we'll be refining what our clinical strategy is. But I think you could anticipate a relatively traditional dose escalation process in a basket trial looking at a wide variety of different tumor types. There is tremendous validation for this target, we believe, in hematologic malignancies, and we're excited about its potential as well in solid tumors. So stay tuned. We'll be revealing more of that strategy and our thinking probably next year as we get closer to when we're ready to enter the clinic.
Excellent. Thank you, Bill. Okay, so maybe just one last one. Obviously, we know Merck acquired VelosBio $2.8 billion for the DLBCL ROR1 ADC. Maybe I see you have data from one PDX model showing some superiority, but just when you design your molecule 003, is that one of your benchmark you try to beat in the preclinical, and also have you seen consistent preclinical data across different models when you compare to your competitors?
Trevor, why don't you go ahead with that, please?
Yeah, Roger, that's not that. What I showed you was the VelosBio linker payload to a different antibody. It's not supposed to be anything like VelosBio's antibody. We were just comparing antibodies with the same properties as STRO-3, but charged with different payloads. So it's a DAR4 MMAE ValCit versus the other exatecan versions. So I mean, I think that what we're looking for duration of response is beyond where the tubulin inhibitors have gone in terms of, or at least to date, and what I've seen publicly in solid tumors. So we're looking to get much deeper responses, longer duration. We're obviously keen to make sure we're seeing what we believe are strong benefits of immunogenic cell death, those sorts of innate cell stimuli caused by sufficiently long and stressed tumor cells that the innate immune system has a leg up to go after. And we think that's going to be particularly important in solid tumors for combinations with things like checkpoint inhibitors. So is tubulin the optimal payload? No, I don't think it is. I think, the data to date with the topo-1 inhibitors has looked really impressive, and that, to my mind, I wanted to ensure that Sutro had the full benefit and was playing on the same level playing field as some of the other opportunities now where the ADC field has gone, and but can bring our own unique differentiation above everything else as well using our engineering platform. Really what we're looking at is how do we get the best possible response with the best possible tolerability for solid tumors, payload, linker, and then the overall design, DAR 8 and where you place them, because they all play with each other to give you the widest possible therapeutic index and the deepest response.
Excellent. Thank you. Okay, that's all for that. Thank you.
Thanks, Roger.
Our next question comes from Nick Abbott with Wells Fargo. Please go ahead.
Hey, great. Thanks for taking my questions. First one, just sort of kind of following up on that last theme. I think you've said in the past, Bill, that for an order of product to become a development candidate, it has to be clearly best in class. So maybe just summarize for us, going back to that Velos Bio product, ADC, what do you think the advantages are over that product?
Trevor, you want to talk a little bit again about the new design elements we're bringing?
Yeah. Hi, Nick. I've had a little look about what's publicly known about the Velos Bio, and it sounds very interesting. And they had a recommended phase 2 dose of 2.5 mg per kg every 3 weeks coming out of their hematological cancers, and I think Merck saw that and probably saw some solid tumor data as well, and made the decision to go. I was interested in a changing in the sort of dosing, the dose fractionation, it seems, late last year, to go for a 3.5 mg per kg dose for solid tumors split into 2 times 1.75 at day 1, day 8 per 3-week cycle. I know the 2.5 mg per kg, they were seeing neutropenia north of 2.25 mg per kg. So I would expect that, trying to push up from 2.5 mg per kg may have some limitations. And I think that the neat way of fractionating the dose like that may give them the opportunity to load up a solid tumor higher to get to higher doses, higher levels of efficacy, while managing a neutropenia response. At least that's my interpretation without knowing anything from what they've done. So neutropenia is clearly an issue there. One of the key aspects that we wanted to do is get out of some of the Cat B related neutropenia liabilities that can occur. And we also noted that although exatecans seem to be very tolerated, they still do have some impact on other tolerabilities as well through the Cat B linker. So we wanted to get away from that just to see what we could do. And so, one notable is that we don't see neutropenia at very high doses in the monkey. Just to give you an idea, we expect to see, if we go from mice efficacy to humans, we think those sorts of exposures, the efficacy is going to be in the 2 to 3 mg per kg range. I hope we don't get too surprised, but somewhere around there for STRO-3. I think in the efficacy on the non-human primates at 45 mg per kg, where we were free of neutropenia and free of interstitial lung disease or pneumonitis type symptoms, at least in the shorter pre-IND non-GLP studies in non-human primates, that translates with exposures to about 15 mg per kg or so. So that gives us quite a wide window from 2 to 3 for efficacy up to maybe 10 to 15 for where you start, where we know we're clean at the moment. But of course, that's not the same thing as doing chronic dosing and where we will eventually go. So, but it's very promising and it's exciting. And the fact that we're seeing an improvement on our own designs for Cat B exatecan going to the beta glue exatecan is even more exciting, because the Cat B exatecans are clinically validated and we do have data points there.
Perfect. Thank you. And then maybe a follow-up. I just want to make sure I fully understand the comments you made about this, cross-species activity, the antibody. Are you saying that it has the same antigenicity profile and that the lack of lung tox is occurring in the context of no or low neutralizing antibody development?
No, I'm not saying that. I'm talking about the ROR1, ability of the antibody to bind ROR1, either rat ROR1 or monkey ROR1. So, it's easy to see good tolerability sometimes when your antibody doesn't bind to anything. And it's the purest form of does your payload fall off and cause some collateral damage. Where it does bind, you've got the added thing. So what mechanism are there low levels of antigen density expressed in some tissue, which normally you wouldn't see if the antibody can't bind to it, but now the antibody can, and it signals that there may be a flag there in that particular tissue. So that's why it's useful to have cross-reactivity to rodents, because you can get an early look without needing to go to monkeys. But in this case, even though the antibody could hit both rodent ROR1, so rat ROR1, as well as non-human primate ROR1, we didn't see any flags there that were indicated there were anything other than known tolerability issues associated with targets. And we were looking very hard, and even though we expected to see lung, we expected to see maybe some neutropenia because of the nature of the payload, and it's been demonstrated before. We didn't see any at these high doses. And so that's testament to the design, the stability, the linker choice, and so on.
Mm-hmm. So just going back to this issue of lung tox, because obviously that would be highly differentiating. I guess how robustly can you test for that outside of a human?
Sorry, Nick, I didn't quite catch your question.
Yeah, so going back to this issue of, the exatecan, I guess, related lung toxicity,
right.
How robustly can you test for that outside of a human? I mean, how confident are you that, you avoid that liability?
Well, you're never very confident. At the end of the day, it has to be in human. But there are some aspects and similarities that we can see from what's published before in what's the patient experience and what's been seen preclinically, which we can draw on. If those relationships are there, then we could make the conclusion that does translate. It might be wrong, but we could. And so improvements like this, I think, are really as we can do in terms of reassuring ourselves that we're not seeing some things that do translate. So, like anything, and of course it's different again because we site-specific conjugates. We're not using cysteine, we're using non-naturals. We're using a different linker compared with Enhertu. Any of that might change and throw our translatability off. But we do these things for a reason. We go into monkeys for a reason. We don't want to go in with an unsafe drug. And we have an antibody that recognizes ROR1, and we're not seeing, it's very well tolerated. We're very excited about going forward. So that's all you can glean, really.
Okay, thanks. And just, I guess that's the last one for me. I know HER2 is not an antigen on your list, but presumably given the data you have, there could well be some interest in a HER2 version, leveraging the beta-glucuronidase linker, an exatecan platform. What are your thoughts about business development and co-development opportunities around this exciting platform?
Thanks, Nick.
I'll let Bill. Yeah.
Thanks, Trevor. We've had a lot of interest from a lot of different partners on both our antibody drug conjugate platform as well as on the IADC platform, not just limited to Astellas. So we think that there are tremendous synergies to be found with a number of different companies that are interested in different targets than we may be interested in, and we're open for business in that vein and looking forward to continuing those discussions. And hopefully today gives people an even broader appreciation of the breadth and depth of our platform technology and the various ways in which we can rapidly advance molecules forward so that we have the best-in-class opportunity, or if people are interested, first-in-class, and be assured of manufacturability. So it's an exciting time for us, and we're delighted to be engaged in so many different conversations. We don't guide, as you know, on any particular deal objectives, but we are actively in conversations as we speak here today.
Great. Thank you very much.
Thanks, Nick.
Thank you. Our next question comes from Boris Peaker with Cowen. Please go ahead.
All right, thanks for taking my question. I'm just curious, is there any competitive data on ROR1 in the horizon that we should be paying attention to or that may impact your development strategy for 003?
I think, Boris, people are still waiting to see how the VelosBio data comes out. That's probably going to be the next most significant bit of data for us to look at. And one of the advantages of being where we are in terms of the development of this asset is we've learned a lot in the field as to how to design better ADCs. We've now got this new linker and this new class of warhead. And so we'll be following, as we have historically, the evolution of programs like the VelosBio one, like perhaps the NBE molecule as well, and really understanding what the opportunity is, what the challenges are, and frankly also what the liabilities are. Because we have such a significantly differentiated molecule, we think we can learn quite a bit and adopt a very fast follower strategy here once we move into clinical development. But I would keep an eye out looking particularly for the Merck program from VelosBio.
Mentioned ADC squared, if that's how we refer to it. When are we going to get an update on what that really is or any kind of programs out of that?
Oh, I'll let Trevor, since he's the one who put out the terminology, handle.
That's just my sense of humor, I guess. But no, it's really meant to capture other systemic, mechanistically synergistic mechanism we might put together. And there are a few of them out there. We're not ready to reveal those yet. But stay tuned.
I think we had enough for everybody today, so we'll just save that for a future date for us.
Got it. All right. Well, thanks very much for taking my question.
Thank you.
Thank you. Our next question comes from Asthika Goonewardene. Please go ahead with your question with Truist.
Hey, guys. First, I want to say thanks for putting this very informative session together. This is very helpful to us, and really appreciate all the detail and color that you provided here. And congrats on disclosing more on 003, which we've been waiting for. Maybe some questions on 003, and also want to get your thoughts on how it compares to the other competitors, the VelosBio and the NBE-02. Maybe let's start with the targeted epitope and the binder. I want to know if you guys how the epitope and the binder that you're targeting with 003 is differentiated from the others. And I got a couple of follow-ups.
Great, thanks, Asthika. Trevor?
Yes, hi, Asthika. There was some background. There was some information around CAR-Ts at one point in certain indications where there was some concern over whether a binding epitope was too far out from the surface of the cells to be retained. In that particular indication, there was evidence that there was some shedding of the ROR1 and the epitope of the particular that the CAR-T would bind to. So, of course, that would be compromising. So we're aware that there are those sorts of discussions going on. That was an immediate concern for us when we embarked on this program. With the VelosBio program, we believe we have an epitope which overlaps. It's not identical, but I think it's pretty. It's gonna. I think it'll perform pretty well, similar to the same in the same epitope as the VelosBio antibody. So from that perspective, I will be watching the VelosBio program very closely and to see whether that is reproduced or whether they see large amounts of dissociation. So we are pretty excited with where we are. We think we've made the right decisions in terms of epitope choice.
Okay. And then just want to confirm, Trevor, the warhead that you're using is exatecan itself, or is it a modified version of exatecan?
The Enhertu warhead is deruxtecan. Our STRO-3 catabolite is exatecan, and the one on the Cat-B linker releases a slightly different catabolite again. So hence this discussion around slightly different potencies and seeing with the different things and slightly different tolerabilities of the ADC in its entirety. We're not sure whether that's driven by the difference in the catabolite or the difference in the linker release or the set of liabilities or whatever. But the bottom line is we're very happy with the choice of the active catabolite we derive from STRO-3 and its linker mechanism.
Okay. And then if you were to maybe benchmark the membrane permeability of this versus, let's say, DXd, would you expect them to be sort of similar, or how do you see those circumventing, Trevor?
We expect ours to be very potent. I haven't done a direct comparison.
And then have you benchmarked the level of immunogenic cell death that the warhead drives versus, let's say, vedotin or hemiasterlin?
I think the topo 1 inhibitors and exatecan itself is known to be a very potent immunogenic cell death stimulus. So I think it's okay. It looks very well. The hemiasterlin itself is really quite good. I think the exatecan's in a even more potent.
Got it. Okay. Just want to try and understand the specific differences and between these different ADCs here. Maybe if I can squeeze in a quick question on MK1484, I just want to get some thoughts on what the next milestone payments have been, or what milestone payments have been recently triggered by Merck taking this into a phase 1 study.
Thanks, Astika, for talking about that asset. We're excited that it's moving forward with clinical development, and when we're ready to talk more about that, we'll be in a position to do so. So I would say stay tuned, but it's not something we're going to be discussing today.
Got it. All right. Thanks a lot, guys. Appreciate all the color.
Thank you.
Thank you. Our next question comes from Ren Benjamin with JMP Securities. Please go ahead.
Hey, thanks, guys, for taking the questions, and congrats on 003 and this presentation. I guess I'm most interested in any sort of combination work that you might have performed, and how we should be thinking about the development of this asset going forward in terms of either add-on combinations or, given kind of what Trevor went through as to how it's easily modified, whether or not you can drop the DAR down to, let's say, 6 and add on, let's say, 2 TLR7 molecules or a TLR7 and TLR8 molecule, essentially get a combination within one molecule. Can you give us some thoughts as to how we should be thinking about that?
Trevor, over to you.
Thought it might be. So, are you talking about ROR1 specifically here?
Yeah, ROR1 specifically. Just
yeah. So it's all about the indication, the antigen combination, of course, and also whether treatment paradigms are standard of care, what sort of tumor we're looking at. Is it cold? Is it hot? Is it one where there's a great deal of resistance to a particular mechanism that you're driving with a payload and so on? So there are already clues precedented, of course, in clinical practice with some of these things, with some of the payloads and different things in combination. We expect the IADC constructs to be extremely good when combined with checkpoint inhibitors. We're providing a component part which is enriching CD8 positive cell populations and driving an adaptive immune response. It would be astounding if we can't reproduce some of the exciting preclinical data we have, that this doesn't actually, if it acts the same way and is driving that bridge from the innate immune cell stimulation or activation through to adaptive immunity, then we would really expect to see a great contribution from checkpoint inhibitors there. And, so those are opportunities to further define and further move. And the fact is we have now a foundation of tolerability that we've begun to build with a DAR8 exatecan ROR1. And all the component parts are pretty well, will be GMP ready in a short period. It's relatively straightforward to switch, say, the light chains with a different non-natural to do as you say, if we so wish. Of course, it's all about money and where we think the opportunity is to generate value for the company.
Okay.
And thanks for giving new research ideas. Right now, we're really focused on driving this asset, the ROR1 ADC, into the clinic as quickly as possible. And certainly the possibilities of combination therapy and or modifications to make this more of an iADC player are downstream. But we want to start with what we think is a very exciting opportunity as an ADC. And with an exatecan DAR8 and this new linker, we think we've got a really great molecule that can benefit patients. But I appreciate the creativity, and we'll be thinking through that stuff as we continue to progress this program.
Got it. As we look at both the low and heterogeneous ROR1 antigen levels, right, like on slide 9, am I reading too far into it? When I look at the ROR1 2+, mouse model, it doesn't seem to achieve the same sort of tumor, anti-tumor activity as the ROR1 1+ levels. And so it would seem to suggest that maybe a biomarker strategy might be important in developing this further in the clinic and maybe selecting against high expressors. Is that too much of a read, or is this more of a mouse model sort of artifact?
Both. I think it is too much of a read. I mean, these are chosen to be sort of representative. I mean, I'm just looking at, okay, have we got good everything? Yes, great. That qualifies a different linker because that was a concern about beta-glucuronidase expression, for example, across human tumors and so on. But I think you're absolutely right. I mean, we would be planning, if we were doing a basket trial in heme and solid tumors, for example, 2 separate baskets, then we will, of course, evaluate whether we need to enrich. And, but I think it is too deep a read on these particular PDX models. They're fraught with lots of other factors going on you can't really quite control for across, by comparison. So I wouldn't read too much from there. To me, it's validating in terms of linker choice and nice to see good efficacy at low antigen density.
Got it. And I guess just one final one for Bill. When do you think, you may have answered this, but when do you think we might file an IND for 003? Would that be next year or the year after?
Yeah, I wish it was this year, but it's not going to be. At this stage, we're working through the process development and the IND enabling work. I think we've not guided to when this is going to be in the clinic. Typically, molecules at this stage, and remember, this is a brand new design for us. It takes about 18 months or so to get into the clinic, but we're going to certainly work hard to beat that. And when we're ready to announce a timeline, you'll be one of the first to hear it, Ren.
Excellent. Thanks very much, guys.
Thank you.
Our next question comes from David Nierengarten with Wedbush Securities. Please state your question.
Hey, most of mine have been asked, but I wanted to ask a question maybe a little more on the heterogeneous expression and what does that mean for cell killing? Is there, or do you think there's a threshold of kind of zero expressing cells and you can still see an effect? And does that change with a general expression level of 1+ or 2+? I know it's kind of a tough question to answer probably in the PDX models, but I'm curious because it might, be reflected in the confidence levels, of tumor types that could be treated, by, Bio3 in the future, looking at, some of the papers of expressing levels and proportion, tumor cells that express and things like that. So I was just curious if you, had any thoughts around, proportion of expressing versus, level of expression in different tumor types.
Trevor, over to you.
Yeah, it's a little too early to know. We do studies where we go from sort of two-dimensional culture to three-dimensional culture, which is sort of, in the cell killing area where I showed you there was no bystander effect, so it's all antigen dependent. That's a 2D culture where any released payload is relatively diluted away. If you go to a 3D culture, you can build up some of the concentrations around there, and you can start to get a feel for how much, primary tumor cell processing there is of a stable ADC to release enough payload to then have a knock-on effect to those that are negative. It's quite complicated. I'm not totally sure it predicts either, but you're right. Somewhere in the middle there's the ideal scenario. I think it's actually rather difficult to. Well, one, I could establish it, but then I know what your question would be of me if I did show you that. You'd say, well, how can you translate? How do you know that? So, it's a no-win game for me. Suffice it to say, there is good it's very potent. There is a good strong bystander effect there. We know the more important thing for me is it can, we're getting sufficient killing of a heterogeneous ROR1 PDX. So it's obviously giving a good, strong response. And we do know from the other things we've done that we are able to ensure that we're not killing immune cells, straight out of sight if those systems are populated. So we'll be doing more than that. I think the designs and efficiency we have with our system allow us to be very, very sparing with the level of payload we're dosing and that reach the tumor, because each payload can efficiently get to the tumor and to the tumor cell. And I think, like with STRO-2, what we're able to see there is good efficacy preclinically, and in MC38 models where you're able to see in combination with checkpoints, we've demonstrated with STRO-2 that we see good enhancement of CD8 populations. There's no apparent clear out because you're using an ADC in some of those systems. You're enriching the tumor for CD8 cells. So there's not such a strong bystander effect that you're wiping out all immune cells, which is one of the disease objectives. So it is a bit of a tightrope though. And our key design is let's get this really efficient. Every internalization event is optimal. Every molecule is optimal. And with that's homogeneous, and it's the right conjugations and the right number for the right potency for the right linker to ensure that every internalization event is optimal.
Thank you.
Thanks, David.
Thank you. There are no further questions at this time. I'll turn the floor back to Bill Newell for closing remarks.
Thank you all for joining us today. Trevor, thank you for such an extensive presentation and discussion. Both of our next molecule, wholly owned, a ROR1 ADC, will be STRO-3. We don't talk about these molecules until we're highly confident of our ability to deliver on them and to bring them to the clinic. So this is a DAR8. It's the first DAR8 from Sutro's platform. It's site-specific with a new and exciting exatecan warhead and a beta-glucuronidase cleavable linker. We also had a chance to talk a little bit today about what our platform is and how we've been building it so that we really have a modular approach with the most advanced linkers, the most advanced warheads, the ability to do dual conjugations, to do mono or bispecific targeting. All of that is really up to the research team here, and they've done a tremendous job of breaking new territory in protein engineering design and chemistry that no other company, we believe, is capable of doing, and certainly not a company nearly our size. And then to actually extend that into an integrated manufacturing system where our biochemical synthesis really works well because we've pre-made many of the component parts, and so we've got rapid scalability, broad flexibility, and a path to commercialization. So happy to really dive more deeply on all of this with others in the coming days and weeks. This is really a summer break for us in a sense that we get to talk about the underlying platform technology and our next up assets. That doesn't mean that we're not hard at work at advancing STRO-2. That continues to move forward, and we look forward to having an opportunity to speak with you all later this year about what our conversations with FDA are, our path forward to a registration-directed study, what our final data sets are, and really what our clinical design is. So this is more of a scientific lecture in some respects and presentation, and looking forward to actually getting back to clinical dialogue with you all later this year. So thanks so much for tuning in today, and appreciate your interest in Sutro and your time today. And that's it, operator.
Thank you. This concludes today's conference. All parties may disconnect. Have a good day.